Well, thank you everyone for joining us. Today, I'd like to share with you what is involved in running the Dovetail micro-C assay in your lab. And that will cover the wet lab side, and it will also cover how you process the data to actually assess whether your library was of good quality or not. So to start off, the Dovetail micro-C assay can be completed in 2 days, and that is a total of 12 hours. It consists of 5 stages and a total of 18 steps. Stage 1 is for sample preparation. This is where you're cross-linking the chromatin, you're digesting, and you're extracting the chromatin fragments. Stage 2 is a QC checkpoint where you're going to assess your digestion. And stage 3 is where the proximity ligation reactions will be carried out. So at the end of stage 3, you would have generated proximity ligated DNA, and that would be day 1. Now in day 2, you're going to take this DNA and convert it into a sequencing library by carrying out stage 4 and 5, which is the library preparation stages, and that would be 4.5 hours. There are several safe stops in the protocol for convenience, and there are also QC steps built into the workflow that will allow to give you the peace of mind that you're on the right track, and also to give you an opportunity to take corrective measures if needed. So this is an overview of the workflow. In the next slides, I'm going to go through the stages one by one. I'll share with you a bit more details about the steps and some tips and best practices. So stage 1, here the chromatin is cross-linked in the nucleus. It's then digested with MNAs. And at the end, you have cell lysis, where you're lysing your cells with SDS to extract the chromatin fragments. In the micro-C assay, we use DSG and formaldehyde for cross-linking in a 2-step cross-linking method. So we start by adding DSG, rotating the cells, then we add formaldehyde, we rotate the cells for 10 more minutes, and at the end, we wash with the wash buffer to quench the cross-linking reactions. So the reason why we use DSG in addition to formaldehyde is that DSG has a longer spacer arm. It's a strong protein-to-protein cross-linker, and it cross-links large protein complexes. So by using it in addition to formaldehyde, we're actually increasing the long-range interactions that we're capturing in the library. So this is a standard tube rotator that we use in-house to rotate the cells. It has a continuous rotation, and the one we use is at 8 rotations per minute. A little bit of tips around formaldehyde and DSG. So formaldehyde is user supplied. It's not provided in the kit. And we recommend that you use a freshly opened formaldehyde solution. So if the solution that you're using has white precipitates, we recommend that you don't use it for cross-linking. DSG, what we recommend is to have the DSG prepared immediately before use and not stored in solution, so DSG can hydrolyze readily. That's why we recommend to immediately prep it, freshly prepared solution, and then use it. In the user guides, we have Thermo Fisher as a recommended vendor for DSG with a part number that takes you to aliquots, pre-aliquoted 1 milligram of DSG. So, and that's the amount you're going to use for the DSG solution for every library preparation. So then you can use a fresh aliquot every time, and you don't have to worry about storing the DSG in solution. And DSG will be solubilized in DMSO, not in water. So now that you actually are done with the stage 1, before proceeding to stage 2, a few more tips about how you're going to achieve this optimal chromatin digestion. The micro-C assay does not require sonication. It's really only relying on the digestion of the chromatin with MNAs. So achieving an optimal digestion is important, and it does influence the quality of your library. To achieve an optimal chromatin digestion, we recommend that you freeze the cells before cross-linking and before digesting. We often get this question whether or not fresh cells can be used. We actually recommend that if you're starting with fresh cells, you're washing them, you're counting them, to then freeze them for 30 minutes at least before you proceed with the assay, because freezing does affect the digestibility of the chromatin. And during development, we have observed that freezing the cells actually will ensure robustness of the protocols to achieve optimal chromatin digestion. Also, cell counting is important because you're going to dial up the MNA's volume based on the number of cells that you're starting with. So to have a more accurate cell count, we recommend that you count before you freeze the cells. And we also recommend that you do your harvesting at low speed spin using a swinging bucket rotor. This will help you also with any cell clumping. So the cells should not clump when you're reducing the spin and using a swinging bucket rotor. This becomes important with some of the cells that are notoriously known to be clumping, like neural cells, for example. So just in summary, you're going to harvest your cells. You're going to wash them with PBS. After washing, you're going to count your cells and aliquot the number of cells you'd like to start the assay with. Our assay calls for 1 million cells. You aliquot it in a tube, spin down, remove the supernatant, and then you can freeze your cell pellet at -80 for at least 30 minutes before proceeding with the rest of the stage 1. Going into stage 2. So stage 2 is more of a QC checkpoint. This stage will allow you to do 2 things. It will allow you to confirm that the chromatin was properly digested before you proceed any further. And it will also allow you to quantify the total lysate yield that you actually recovered from your cells. So how do you do that? This is the total lysate that you have recovered after cell lysis with SDS. You're going to take an aliquot, 2.5 microliter aliquot, to run stage 2. And the rest of your lysate, you're going to store it at -80. This is the lysate that you're going to be using to proceed further to stage 3 in the protocol. So your aliquot is essentially cross-linked chromatin. The first thing you're going to do is reverse the cross-links, degrade the protein using proteinase K treatment. You're going to then purify your DNA. And at the end of the purification, you're going to run that purified DNA on an analysis instrument. You can use tape station, bioanalyzer, or a fragment analyzer. So these are 2 examples of the digestion profiles. This one is run on a fragment analyzer. This one is run on a tape station. So we're going to start with this one first. This profile, you actually can see this peak represents the mononucleosomes. This peak represents a dinucleosome. These are the trinucleosomes. So the profile we're looking for has around 40 to 70% mononucleosomes. How do you determine this percentage? You will actually create a region that includes that first peak, which represents the mononucleosome. On the tape station, that first peak usually runs between 50 to 250 base pair, but that's not always consistent. So really what you want to look for is a region that includes that peak. So from this dip or valley to this dip or valley. And when you create this region, a region table is automatically generated under your electropherogram, and it will output this percentage of total. So how much of your total DNA is between 50 to 250 base pair? You want this percentage to be between 40 and 70%. In this case, it's 52.22%. So this chromatin is optimally digested. This profile passes QC, and you can proceed with stage 3. Looking at the profile assessed with a fragment analyzer, it's the same thing. This is your mononucleosome, trinucleosomes. And here the smear analysis, also around 50 to 250 base pair, so capturing these 2 values, indicates 48.4% total. That is between our window of 40 to 70%, and this digestion profile passes QC, so you can proceed further with the protocol. So what happens in the scenario that you have an over or under digested chromatin? An over digested chromatin is like this case, where you don't really see much of the di and trinucleosomes. Your mononucleosome percentage is above 70%. And your underdigested chromatin is where you have your percentage of mononucleosome is less than 40%. You don't really see much of that peak, and you see quite a large tail having larger fragments. So if you are over or under digested chromatin, we don't recommend that you proceed with a protocol. We actually recommend now that you take a corrective measure and to optimize your chromatin digestion before proceeding. And we do offer guidelines for how to do that and help into optimizing your chromatin digestion profile. So now that you've actually confirmed that the chromatin is properly digested and that you quantify the total lysate yield that you have, so for that, you will just take your purified DNA, the same DNA you ran on tape station, and just quantify it with qubit. So when you quantified your DNA and confirmed that the chromatin is properly digested, you can proceed with stage 3. For stage 3, you're only going to proceed with 1,000 nanogram of your chromatin. So if you have less than 1,000 nanogram, you're going to use everything, all the lysate that you have. If you have more than 1,000 nanogram, you can store the remaining of your lysate back at -80. The first step in stage 3 is binding the chromatin to chromatin capture beads. All the proximity ligation reactions will take place on the beads. The second step, you're going to repair the ends and make them adaptable to or compatible to receive the bridge that you're going to ligate in the third step. And this bridge is a custom-made oligo that has a bite and tag on it. And the way that we're going to have the proximity ligation occurring is you have 2 fragments that are cross-linked in the 3D space. Those fragments should have received a bridge or that custom-made adapter at their ends. And that's how they're going to be able to ligate together in order to create this proximity ligated DNA molecules. So you have fragment 1, fragment 2, they're cross-linked, and they're ligating to each other through both having received at their end the bridge that it has a bite and tag on it. So now that you have proximity ligated your DNA molecules, you're going to reverse the cross-links, degrade the proteins, and then you're going to purify your DNA. And for this DNA purification step, you're just going to use a standard bead cleanup with SPRI-select beads. This DNA will then be quantified, and that's the second QC checkpoint in the protocol. And we expect that you're going to recover around 450 nanogram of DNA. So you start stage 3 with 1,000 nanogram, and you ended usually with around 50% recovery of the DNA you started with. This is important because you're going to proceed with the library preparation, the last 2 stages of the protocol, with only 150 nanogram of that DNA. So whatever you have remaining, you can store it at -20. And this will be helpful for you. Let's say you do want to include a technical replication in your project. You can use that DNA to prepare the library, the second library. You don't have to start from the beginning. And also, let's say that your project calls for more depth and more libraries. This is when, again, this DNA will come in handy. We do have 2 safe stops in this stage. So the intra-aggregate ligation and the cross-link reversal reaction can both be incubated overnight. That's only for convenience. Those 2 reactions are complete at 1 hour, but for convenience, you can leave them overnight. The other thing I wanted to mention is all of those reactions, you're actually going to be putting your tube shaking at 1,250 RPM. We placed our tubes in an agitating thermal mixer, and I just wanted to show you the one that we use in-house. It's a standard thermal mixer, and it has the heating and cooling features as well. So now that you have generated your proximity ligated DNA, you're going to take that DNA and convert it into a library by running stages 4 and 5. Starting with stage 4, the library preparation, this is really standard NGS library prep. You're going to repair the ends, add an adapter, and then purify your adapter ligated DNA. To reiterate, the library preparation does not require sonication, so you're just going to take your proximity ligated DNA or the DNA from end of stage 3 and proceed directly with end repair. In order to do these stages, you're going to need the Dovetail library preparation module that we offer as an add-on module. The protocol as written is actually specific for the Dovetail library preparation module and the primers that we supply. If you have a library prep module that you use routinely in-house, you can always reach out to us to see if that's compatible with our assay and how you would integrate it with the assay. So now that you have purified your adaptive ligated DNA, you're going to proceed with the last stage of the protocol. And before setting up your PCR amplification, like you would do with an NGS library, we actually include one step, and that is for streptavidin bead pull down to enrich specifically for the proximity ligated DNA molecules. So you're going to bind your adaptive purified DNA or adaptive ligated DNA to the streptavidin beads. You're going to do a few washes to remove anything that did not bind to the beads. In this case, anything that was actually not biotinylated. The biotinylated DNA molecules are now on your beads, and that's your template DNA. You're going to set up your PCR amplification reaction on the beads, and then you're going to get your amplicon, purify it with a bead cleanup, and do a size selection with Spry Select beads as well. We do a double-sided size selection, so we remove anything less than 350, anything above 1,000 base pair. And then we have your library that you can run on a tape station or a bioanalyzer or a fragments analyzer to assess the size distribution. And we're expecting the library to be between 350 and 1,000 base pair. So once you look at your profile, you're going to see some of these bumps. This is actually normal. It's reflective of the chromatin that was digested with MNAs. So it's reflective of that digestion profile that you had. One more thing about this stage, the reason I mentioned in the previous slide that if you're not using the Dovetail library prep module, please reach out to us to see if your library prep kit is compatible with the assay and how to integrate it. It's really mostly because of that PCR amplification step. The PCR amplification step is set up on the beads, and not all polymerases and enzymes are compatible with the streptavidin beads to amplify your template DNA. So now that you have checked your size distribution, you can also quantify that library, and you're going to be ready to proceed further with sequencing. The library can be sequenced on any Illumina platform, and I'll speak a little bit more about the sequencing parameters in the next slide. But with this stage, you actually have completed your library preparation. So in terms of sequencing, since this is really a Hi-C based method, you're going to need to sequence your library in paired end mode. For the read length, you can sequence your library as 2 times 75 base pair, 2 times 100 base pair, or 2 times 150 base pair. We don't recommend sequencing with a read length that's less than 75 base pair. And that's because of the aligner that we use to process the data later. In terms of depth, before you proceed with deep sequencing, we actually recommend that you do a shallow sequencing of 1 to 2 million read pairs. And that is to run a QC analysis and check the quality of your library before deep sequencing. For deep sequencing, the micro-C library can be sequenced for a maximum of 300 million read pairs, which is typical for a Hi-C library, and that would be a Hi-C claim. So in terms of what is the number of library, what is the depth or the coverage that you need to achieve with a micro-C library, we often get this question, and that really is dependent on what is your research goal. So if you're looking to call AB compartments, for example, you're really only looking for a 1 megabase resolution and a 3x coverage. For that, you only need a 30 million read pairs, and that's, all these calculations are based on the human genome. So you need 30 million read pairs, 2 times 150 base pair. that is achieved with one library. If you are calling TADS, then you're looking more at a 50 KB, 25 KB, or 10 KB. That's a 30x coverage that is achieved with 300 million read pairs. And for that, you need 1 to 2 libraries. You can do with one library because you can sequence your one library to 300 million read pairs, but it's going to depend on your library complexity. And we'll talk a little bit more on this when we talk about how you will assess the complexity and the quality of your library. So you might actually need 2 libraries for that. For loop calling, which is mostly the main application for a micro-C assay that we see amongst our users, you're targeting a resolution of 10 KB or 5 KB. That's an 80x coverage. You need 800 million read pairs. And for that, you actually need 3 libraries, each sequence to around 300 million read pairs. So how do you assess the quality of your library? To do that, I will walk you through the workflow that we have on our Read the Docs. But before, I'd like to stop and just answer the question of what is considered a valid read pair in an MNase or a micro-C assay. So this illustration shows you the cross-linked chromatin fragments that were digested. And then you have 3 different ligation events that can take place. You can have a self-ligation event, which is here this red fragment will ligate on itself and will form a circularized molecule. You can also have religated events. In this case, those 2 fragments were cut at these sites. They ligate back at the same sites they were cut from, and that is a religated event. It's also known as, an invalid read pair. The last ligation event that can take place, which is the valid ligations, this is a proximity ligated event, which means that these 2 fragments were actually physically not next to each other, but they were cross-linked or co-localized in the 3D space. And that is the ligation that you want to have. So those self-ligated molecules or circularized molecules will not make it into your library because we do not sonicate. So you can't turn this into sequenceable molecules. The only 2 ligation events that you'll see in your library would be religation and valid ligation or proximity ligation. So how do you tell from the read pairs that you're seeing if these originated from an invalid religation event or they're valid from a proximity ligation event? That's going to depend on your insert size for your cis read pairs. So at the end of your stage 5, you actually size selected your library to be between 300, 350 to 1,000 base pair. So the largest molecule you have in your library is 1,000 base pair or 1 KB. So that means if you have an insert size that is essentially the physical distance that is spanning, that your molecule is spanning, that is larger than 1 KB, the only way this could happen that you have a physical distance that's larger than your largest molecule is if that originated from a proximity ligated event. So that would be considered a valid read pair. So looking at these valid read pairs or this read pairs, if you have a read pair with an insert size that's between 350 and a 1 KB, this is just a religation event that happened. If you have an insert size that is more than your longest fragment, that means that it's more than 1 KB, this must have originated from a proximity ligated event where the fragments were not physically next to each other, but they were co-localized in the 3D space. And that's how you would bin your read pairs as valid versus invalid read pairs. And by the same logic, a trans read pair are always considered valid read pairs because they are spanning 2 different chromosomes. So it's good to have this in mind as we talk more about how do you actually process your read pairs to check the quality of your library. So the first step is you're going to align your raw FASTQ sequences to a reference genome using BWA-MEM. Because we're using BWA-MEM, you don't need to do any trimming for your read pairs to remove the adapter or anything. You don't have to do any pre-processing. You would actually just start with the raw FASTQ files that you have and align them with BWA-MEM. we use this -5 SP flag. And what this means is that you're going to start the alignment from the 5' end. When it will get to a chimeric event or a bridge, it will actually stop, have a soft clip, and it will start from the 3' end. So it will first treat the 5' end alignment as a single end alignment, and then it will start from the 3' end as a supplementary alignment. So what you're really doing is you're doing 2 single end alignments in one command, and at the end, you're going to have all the alignments merge in one BAM file. And that's why we're using this flag. So after you align your FASTQ files to the reference genome, you'll actually be able to filter out the read pairs that did not map or did not align, the low mapping quality read pairs, and here we have the quality score at 40. So these would be the read pairs that mapped with a mapping quality score less than 40. And you're going to have your mapped read pairs, which are the read pairs that mapped with a high mapping score, which is a mapping quality score above 40. So those mapped read pairs will then be piped into pair tools, and pair tools will flag the PCR duplicates. So what you're going to end up with after this first step, you're going to have the counts of the read pairs that you got off of the sequencer, so what you started with, how many read pairs are unmapped, how many read pairs are mapped with a high mapping quality score, and how many read pairs were PCR dupe versus no dupe read pairs. So in this table, and that's a table you get when you actually run the workflow that we have on Read the Docs, you're going to get one column that's a count and the other column that's a percentage. And in this case, the denominator is your total read pairs. This is another way of showing the same data just through a graphic. So this is your total read pairs, that's 100% your denominator. Out of that, you have 82% that were read pairs that mapped with a high mapping quality score, around 14% mapped with a mapping quality score less than 40, and 4% were unmapped read pairs. From the mapped read pairs, you have 0.14% that are PCR duplicates, and around 82% that were not duplicates. So that would be the first step, and then you're going to proceed with a second step with your mapped read pairs, the high quality read pairs. And now you're going to ask the question, are these read pairs cis or trans? And are these read pairs originating from a valid proximity ligation event, or are they invalid, originating from a religation event? To do that, you're also going to use pair tools. So pair tools can classify a read pair as cis, so spanning or mapping to the same chromosome, or as trans, so mapping to different chromosomes. And that's how you're going to have your cis read pair and trans read pair number. The trans read pairs are valid read pairs. But for the cis read pairs, you have to ask the question, what is the insert size? Is it less than 1 kb and more than 1 kb? So what pair tools does is it generates a histogram with the counts of read pairs at different insert sizes. So you can apply a cutoff wherever you ask pair tools to. And in this case, we had the 1 kb, which is our largest fragment size. So you have the counts of read pairs less than 1 kb and the read pairs that are above 1 kb. You can see that the count actually, the further out you go, the count drops, but the sum of your read pairs is large. So here it's showing that you have read pairs less than 1 kb is around 100,000 count, and the read pairs that are more than 1 kb is around 1 million. So again, this is a count and you get the percentages here. In this case, the percentages, we're taking the no dupe read pairs that are the mapped high quality no dupe read pairs at the 100%. So this is in this case your denominator, because this is really the useful data that you want to proceed with. So what this is showing is that you have around 28% of your trans read pairs. These will go into the valid read pair bucket. And you have around 71% of cis read pairs. These will be categorized as less than 1 kb, which is this bucket here, that's invalid, or more than 1 kb, which is considered within the valid read pair bucket. We're also showing the number of, or the count of read pairs that is more than 1 kb. And that's to give you another depth of what type of or what percentage of long-range information or enrichment of long-range information you've captured in your library. So this is to assess the long-range information or the heisiness of your library, but you also would like to assess the complexity of your library and how much, to what depth you can actually sequence that library. And to do that, we use pre-seq tool. So what pre-seq does, it will look at the number of duplicate read pairs you have in your shallow sequenced library. And it's really only valid and applicable for a shallow sequenced library. If you've already deep sequenced your library, the pre-seq tool is not really relevant. So it's going to look at the number of read pairs in your shallow sequenced library, and then it's going to subsample the data to look at what is the frequency of these read pairs that are duplicates popping up. And then it will create a model, and it will extrapolate the number of unique read pairs that you're going to get at a given depth. So you have it at 300, 500, and above. We look at the number of unique read pairs that you can get as an estimate for 300 million read pair depths, because really that is the depth that you would sequence, or the maximum depth you would sequence your library to. In this case, you have 246 million unique read pairs at a 300 million read pair depths. That would be considered a high complexity library, and that library you can deep sequence to 300 million read pairs. But you can have two different scenarios, one which is a medium complexity library and one that's a low complexity library. If you have a low complexity library, so you have a very high percentage of read pairs and a low estimate of unique read pairs at 300 million depth, we don't recommend that you actually deep sequence that library, because that's not money well spent for sequencing. You're not going to get a lot of informative read pairs out of that library. For that, we would recommend to prepare another library, and we can troubleshoot why the library was of low complexity. If you have a medium complexity library, this is where really having a shallow sequencing step to assess the quality of your library before deep sequencing becomes particularly helpful, because you can take some measure in this case, and instead of deep sequencing one library to 300 million read pairs, we'd recommend that you prepare a second library, pull both libraries together, and deep sequence to 300 million read pair from the combined pool. And in this case, you don't actually have to start the assay from the beginning. You can go back to the stage 3 DNA that you had in order to prep from that DNA the second library and pull both libraries together for 300 million read pair depth. So that would be the last step in how you assess the quality of your library, which is mainly assessing the complexity and the enrichment of long-range information in your library. All of what I discussed is available for you as a self-serve document. So we have a document that you can access at this link that walks you through what each metric means and how it was computed. And we also have Read the Docs workflow available that is basically you can take the command, copy and paste them, and run your data to process them. But we also have control data sets that you can take in order to just have some practice with running the QC workflow. So these will be available at our website and will also be available on this slide deck that I think from Nikki, you're going to have it as well available to you. That was what I wanted to share from my end. And now I'm happy to take any questions you have or have any discussion about the micro-C assay and how you would set it up in your lab. Let me just orient myself. So I'm going to go first to the chat box. And then we have the Q&A box as well. I'm seeing questions in both. Okay. Okay. So the question is, what maximum percentage of BCR dupes is acceptable? So when you're doing the shallow sequencing and running the QC analysis, What would be acceptable for a 1 to 2 million read pairs is anything less than 1%, because that is really, that is a good indicator that when you deep sequence your library, you're not going to have a high percentage of dupe rate. So 1% or less duplication rate for a 1 to 2 million read pairs would be what we considered, a high quality or high complexity library. Another question is, should you flash freeze the cell pellet before starting the assay? So at the freezing step, you can actually freeze the cell pellet just by placing it at -80, or you can freeze the cell pellet by flash freezing it first in liquid nitrogen and then placing it at -80. So it's really up to you. In-house, we just take the cell pellet and we place it at -80. Another question, how long can I freeze the cells for? Can I freeze them for longer than 30 minutes? Yeah, so 30 minutes would be the minimum duration or that you should freeze the cells for. But actually, if you have cells that have been frozen for months, 6 months, or even longer, you can still use them as long as they were not subjected to freeze and thaw cycle. So you really want to start with a high viability sample because the viability of the cells will also affect the library quality. A question about optimizing the digestion. How should I optimize the digestion? So to optimize the digestion, we usually keep the temperature and incubation time the same. And what we do is we just change the volume of MNAs. So based on the profile that you get, if you are if you have underdigested your chromatin, then we would reduce the volume of MNAs that is used. If you have overdigested your chromatin, if you've underdigested your chromatin, we'll increase the volume of MNAs used. If you've overdigested your chromatin, we'll reduce the volume of the MNAs that is used. And we're happy to help. If you would like to share the profile with us, we can give you more guidance on how much to decrease or increase the volume of MNAs by. So my microC library or the bioanalyzer didn't have distinctive peaks like the one you showed. Why is that, and how can I troubleshoot? I think the best answer here would be if you don't mind sharing the profile with us, we'll be able to have better advice. So I would say if you can share the profile with us at support@cantatabio.com, we'll be able to have just a more curated advice. But in general, when you don't see a distinctive peaks, I think you mean you don't see the typical MNAs laddering of mono, di, and trinucleosome. Usually it's either because the chromatin is very underdigested, or because the chromatin is very overdigested. The bioanalyzer should give you, in general, the same profile that you get with a tape station. The only difference between the two that we see is that on a bioanalyzer, you might see a small peak around 65 base pair, which is like sub-mononucleosome that we don't really pick up on a tape station, and that's not an issue at all. But you should still see on a bioanalyzer those MNAs laddering. So if you don't mind sharing the profile with us, and I'll also, if we don't hear back from you, I'll reach out to answer the question. Another question, if you can use Hi-C Pro to process the data. So we don't actually recommend using Hi-C Pro. We know that it's commonly used for Hi-C data, but Hi-C Pro uses Bowtie as an aligner, and Bowtie chokes on that bridge event that we have. So when Bowtie is trying to align the read pairs that have that bridge ligation event, it doesn't know where to map them to and then flags them as unmapped. So you're going to have a underestimate of really how much useful mapped read pairs you have in your library. Whereas BWA-MEM will actually deal with this bridge with the soft clipping. So it will still map your first read and then align the read pair from the 3' end again as 2 single end alignment. So we've seen some of our users who are really keen on using Hi-C Pro. What they do to go around that bridge ligation is they actually trim They're FASTQs, and sometimes they trim them up to 50 base pair to reduce the probability of having that bridge ligation, and then they can use high CPRIP. So a question about, could I use nuclei instead of cells as starting material for micro-C? So the question here, the answer here really is, we've used in-house mostly cells. We have experience using nuclei, but that's coming more from plant tissues. And we use nuclei from plants because we try to reduce any impurities that you can have in a plant. For cells, we usually just use the actual cells, not nuclei. That's not to say this is something you cannot try, but that's not something we've routinely done in-house. For the nuclei, if you'd like to try as starting material, you would still start with frozen nuclei. So you can flash freeze your nuclei in this sense, and then start the prep. The only thing you would have to dial in might be the amylase volume that you're using for this nuclei, depending on what chromatin yield you would expect from them. So for cells, for example, we start with 1 million cells. You want to see if that transfers to the nuclei to start with. Another question about tissue. Can I use tissue to run the micro-C assay? So our micro-C assay has been validated for mammalian cells, but we do have guidelines and R&D protocols available for our users who are interested in other than cells. So we have for mammalian tissues, for Drosophila, some plant samples, as well as yeast. For the mammalian tissue protocol, the main difference would be in the sample preparation. So that would be stage 1. The rest of the stages, stage 2 to 5, there is the same across any sample type. So whether you're using cells or tissue, you're going to follow the same stages. But stage 1 is different for tissues, and it will involve grinding the tissue in liquid nitrogen. And after you grind the tissue, you'd want to cross-link it, and then you will filter the tissue by running it through 200 micrometer and 50 micrometer filters that we supplied. And this is really, you're trying to isolate the cell or nuclei-sized material for the enzyme to be able to access the chromatin. So after filtering, you start setting up your digestion and the cell lysis, and then you continue from there. So really the main difference would be that you need to include grinding and a filtering step for tissues. And the amount of tissues to start with would be around 20 milligram. That's usually if you're talking about a brain tissue, liver, kidney, something that we know has high cellularity content. But when you're working with more of a muscle tissue, like a hard tissue, stomach, skeleton, you will need to start with a little bit more, because that 20 milligram usually doesn't give you enough chromatin yield. So you need to start with a 50 milligram. But if you're interested in the tissue preparation, please reach out to us at support@cantatabio.com, and we'll be able to provide the sample preparation protocol that we have for tissues. Is it possible to get a copy of your slides, or will a replay of the webinar will be available? I think you'll be able to provide both. The copy of the slides and the replay of the webinar will be available. Nikki will be better person to address this question, and I think she is typing an answer. Just looking at the more questions we have. I have a question that I used hiccups and arrow tools to extract the CHADs and loop domains. I got many cis contacts, but I don't see trans contacts between different chromatins. Could you please help with this? Thanks a lot. Yes, so I think with this question, if you don't mind emailing me I'm going to just add the my email address in the slide deck. And I think when Nikki will share with you a follow-up email to this webinar, we'll also include the support inbox. For now, it's support@cantatabio.com. So if you don't mind reaching out with the different output that you had, that would be best, because then we can really address the question with a little bit more information. But for now, with not seeing the trans content, I think the first thing we would want to do is just, you'll definitely would have had trans read pairs in your library. So it's only about how we're visualizing that trans space versus cis space. But I will answer this question better offline with a bit more information. So if you don't mind reaching out to me, or if I don't hear back from you, I'll reach out to you as well. We can have your email address. I think we have one more question that we're able to address, and then the rest of the questions, we might address them offline. We'll send you a personal email to address your question. And that question is, for the tissue preparation, do you supply enough reagents to optimize the digestion? So we actually have, for both the cell preparation and the tissue preparation, the reagents that you're going to get that are used for the digestion optimization. So for stage 1, you're going to get those in excess to allow for any optimization that is needed. The digestion is highly reproducible between different, within the same cell type. So if you have optimized the digestion for a specific cell type, and if you want to process more aliquots, maybe to have biological replicates, the digestion should be highly reproducible. The time when you really need to optimize that digestion would be if you're starting with a different cell type, and that cell type would require more enzyme. So for example, we have GM cells, for instance, would require 0.5 microliter for 1 million cells. But if you're working with fibroblasts or with cancer cells where you would expect a more higher yield, then you need to increase the MNAs to 1 or 2 microliter. But once you do that for your cell type, you can use the same volume and it's reproducible. But yes, we do have enough reagents to allow you to optimize your digestion. And you can reach out to us as well if you need any help on the other pair of eyes to look at your digestion profile. We're happy to do that.